Thank you, Dr. Zhang.
Thank you, Tracy, for the kind introduction. So good morning. I'm going to talk to you about a group of very unique cohort that infected by SARS-1 in 2003 and then followed by SARS-2 infection in 2023. So in the interval of 20 years, we wanted to understand what our immune system, particularly the memory B cell response, and what's the natural status, and then what the implication of those findings for our antibody drug development and the next generation of vaccines. So my laboratory has been studying the pathogenesis of human viral infections, particularly focused on HIV, and with the advent of this new technology, we rush into the coronavirus studies during the COVID pandemic. So the ultimate goal is to study the pathogenesis and then using the antibodies and antigens that evolved and later on designed for the next generation of vaccines. So as Tracy just mentioned to you, in collaboration with Zhang Zhen from Shenzhen No. 3 Hospital during COVID, since China was hit first and then the first victim of COVID, we took advantage of those situations. We studied a lot of basic immunology, particularly involving the antibodies and antibody responses during natural infection. And in collaboration with a company here in China, also National Institute of Health in the US, we will show that the combination of two antibodies has demonstrated 80% efficacy in reducing hospitalization and deaths, and approved by China FDA to treat COVID patients and saved many lives during the pandemic. However, as Zhang Zhen illustrated to you, that whatever type of vaccines that we have, we continue to be infected by the wild-type COVID, but also by the variants of COVID, largely due to the immunity induced through muscular immunization fail to reach our upper respiratory tract. That lower level of immunity has open and vulnerability result in this vicious cycle of infection and escape. So many of our immunity are the root cause of this selection, and then result in many variants that illustrated to you by Zhang Zhen. So this is where we are with this rapid and vicious cycle of mutations. As you could realize, that published by many lay media and the scientific publications. So we are at the age of KP 0.3.31. And that luckily not associated with the more hospitalization and the deaths, but has escaped many antibodies and also vaccines immunity. So in light of this very complex immune background, so what are those means? So what are the immunological consequences of those complex infections, vaccinations, and breakthrough infections? And what are the implications for the next generation antibody drugs and vaccines? So we try to answer these two major questions because it's critical for our next generation vaccines. Certainly, there's a very active vaccine development activity, particularly focusing on our upper respiratory tract here in China and also in the United States and the rest of the world, because you have to build a barrier to stop virus at its site of entry. That being said, but it's very challenging, not many vaccine has been developed using the respiratory route. So in light of these two major scientific questions, we were very fortunate to have a 5 cohort of individual that's very unique around the world. Some are primary infected, and some are breakthrough infected. Here highlighted by orange and green. So as you can see, we have the first cohort that is the SARS-1 primary infection cohort that was enrolled 20 years ago when SARS outbreak occurred in Shenzhen, China. The second cohort is the primary SARS-2 wild-type cohort that in collaboration with Zhang Zhen collected during the early phase of SARS-2 pandemic. Very valuable and then very authentic. The third cohort is a BF7 primary infection cohort that was enrolled during the China releases control policy at the end of 2022 and early 2023. Most importantly, we have this number 4 cohort that's infected by SARS-1 20 years ago, and then 3 vaccinated with the inactive vaccines, and then breakthrough infections by BF7 variants, which was very prevalent in China, in Beijing, and in most part of China. The 5th cohort is the vaccinated by inactive vaccines, and then breakthrough by BF7. So with this 5 unique cohort, it's really offer a unique opportunity to study our B-cell responses, what our immunity look like against various of variants of SARS-2 as well as SARS-1. So Zhang Zhen has already illustrated to you very nice background information, so I don't need to delve into so many details. But I'd like to show you a very real SARS-CoV-2 virus under the tomography done by Professor Li here at the Tsinghua University. So this is a real SARS-2 viruses. As you can see, like many other viruses, look like a crown. And then the spike protein on the surface of the virus are the major neutralizing antibody, are the target of neutralizing antibody. Because of these variations that enable the virus to escape from many antibodies. So what we have been doing is we generate a panel of pseudoviruses carrying the envelope from various major variants, and then to study our sensitivity to serum collected from the 5 cohort of specific SARS-1 and SARS-2 infections. So what do you see? The upper is the primary infection, the serum from a primary infection. The lower panel is from the breakthrough infection from those 5 cohorts. So the take-home message is, as you can see, during primary infections, and then you can see it's very specific to its own viruses, like here in red, the SARS-1 infected individuals are mostly neutralizing towards its own viruses or very closely related strain, which is not surprising. Green is the wild-type SARS-2 viruses against very close resembled envelope, but is losing neutralizing activity to the subsequent variants. BF7 is the blue one's primary infections generated against its own, but also have fairly broad neutralizing activity, suggesting different viruses have different antigenicity, which is very, very clear and which is also inexplainable because the viruses vary several antigenic sites, but how come that induce such a broad or different immune response? But put that matter aside. If you compare the breakthrough infection that had either primarily infected by others or vaccinated, you could see the neutralizing breadth improves substantially. And then the magnitude of antibody response also increased. So this is something argue for the boosting vaccination against whatever that strain is being circulated. The most interesting graph that we generated so far is taking advantage of those serum neutralizing titer and then convert that into antigenic map. So on the left panel, you could see we have, based on the neutralization activity, we could classify all those variants into 4 major serotypes. One is the wild type cluster, particularly together with RATG that Zhengli Laoshi presented yesterday. And that was a very original SARS-CoV-2. And SARS-CoV-1 is up there, it's very, very far. And then the serological distance is about 4.47. And then the BF7 breakthrough, but this is a primary, we forgot, primary serum activity is the most revealing, because when you have a breakthrough infection, your serum basically messed up because there's so many variants. So you can see BF7 is up here, and then JN1, XBB is all here. So you have basically you have a 4 serotype, if we could call it. But most interestingly, if you are breakthrough infected by BF7, and this group, the distance is shortened, so they become a cluster together. And if you break through by BF7 in SARS-1 and vaccinated individuals, you shorten the distance from here to here. So all this become one group. This is the antigenic map that have not been shown so far because they don't have the luxury of primary infected serum. So what this tells us? This tells us that the breakthrough infection really help, we hypothesize, really help to generate antibody that may have a better breadth. Otherwise, it wouldn't cluster together, be able to recognize so many different variants. So in that sense, breakthrough infection doing a fairly great thing for our immune system, because despite of the clinical manifestations or syndrome that we experienced or we endured, but the antibody generated really help us to have a breadth, which is very stunning. But whether this finding can be confirmed at the molecular level. So what we did was, over the past 15 years, since I've been in HIV field for long, we have developed a very high throughput monoclonal antibodies isolation and characterization platform. So we're able to pick the single B cells from peripheral blood or the lymphoid tissues to get as many antibody as possible during early COVID as well as later on for this cohort. So I'm going to go flash quickly about those antibodies. In collaboration with Zhang Zhen, we collected over 1,000 SARS-2 monoclonal antibodies from the primary and breakthrough infected patient. In collaboration with colleagues from Peking Uni Medical College, we collected over 100 SARS-1 primary infected patients. In collaboration with Professor Li at the Peking Uni Medical College, we also have over 650 SARS-1, SARS-2 map. But when you put those data together, what does it tell you? It tells you in those group patients infected 20 years intervals, there's a 61% antibodies able to recognize the SARS-1 protein. And then is 39% is able to recognize SARS-2, but not SARS-1. So you can see, despite of this 20 years interval, we still have a very strong imprinting effect on what we saw 20 years ago. But leave some 40% space to develop the newer antibody against SARS-2. So that's a fascinating discovery, because our memory B cells are really long. That's 20 years. And then for this 1 year interval, once you have vaccinated and also breakthrough infected, almost 99% of your antibody recognize your vaccines. There's only one antibody that are able to recognize the breakthrough infection, BF7 specifically. So suggesting the shorter the interval, the magnitude, or the stronger your memory B cells exist. So your memory B cell exists not depending on the time interval, but also depends on the antigenic distances, because your initially for this group is SARS-1, and later on is SARS-2. It's fascinating memory. And then we break it down to those antibodies to different class to see where they recognize on the spike protein. This is a $1 million slice because it's really combined a lot of information, but extremely revealing. Let me walk you through in the last couple of minutes. So if you see the second line first, this is the antibody that we obtained from the SARS-1 infected patients 20 years ago. So basically, the majority of the antibody that recognize receptor binding domain, we could classify them into different class based on the antibody competition. So you could see they recognize they belong to class 1, class 2, class 3, class 4, class 5, but they are neutralizing activity only to itself, majority to SARS-1, 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 SARS-1. A little cross-reactivity to wild-type SARS-2, BA7 variants, and BA5.2 variants. Minute level of cross-reactivity. However, 20 years later, breakthrough infected by SARS-2, all this antibody we isolated not only recognize class 1, class 2, class 3, class 4, class 5, but their neutralization breadth expand to SARS-2, all these variants. Amazingly, that our antibody response not standing still, but began to expand its horizon. That's very, very revealing. And then if you apply the same technology to study the primary infection of wild type 2 that Zhang Zhen we collected during the early. So all those early strains are quite specific to wild type SARS-2, and a little bit of cross-reactivity. But if you break through infected by BF7, those antibodies not only recognize them, but also began to expand to the neighboring classes. So it's amazing capability to go around the antigens and then cover much more bigger space of the antigen. Whether that is true or not. So we get all these representative antibodies, and then in collaboration with Professor Xinchen Wang in the audience, we have crystallized many representative antibodies suggesting this is a class 1, class 3, class 5. We using crystal structure to confirm this is real. And then most importantly, the expanded antibody not only able to bind to the antigen, but also to demonstrate much higher neutralizing capability. So our immune system is amazing. We are not waiting there to be killed by the virus. We are expanding our arsenal towards other regions, while still optimizing, treating the region that's really dominant before the breakthrough infections. And then we did animal study. Many of the expanded antibody also bear a potent protection against infection, against transmission in the animal model, hamster model. So now through this structure and the function studies, we have a clear idea where the antibody recognized, where the RBD variations is, and then you could see the early days, many of the antibody recognized here, but because of the mutation escape, our antibodies began expand to those wide area that's much less variable. Although there are still some struggling to focus on there. So it is amazing expansion of our immune system. So what are the immunological consequences and what are the next generation vaccines? Basically, what we're seeing, partially due to the epitope masking of the antigens, and then partially because of our immune memory is so strong, it's not able to recognize a new antigen that come later on. So one sentence, what that mean for our vaccine development? I know in many countries have already developed the bivalent vaccines, wild type BA1, and then now XBB. So if this story is true, then in fact, regardless of what vaccine you use, you're going to expand our antibody horizon anyway. So in theory, this may be more provocative that we don't need to catch the virus. You just keep injection, maybe the old vaccine, that would be enough. Okay, so with that, close my presentation, and happy acknowledge my group at Tsinghua University, Hong Kong University, a long-term collaborator of Shenzhen No. 3 Hospital, Zhangzhen in particular, and my long-term collaborator, Professor Li at Peking Union Hospital, and then Xinquan Wang at Tsinghua Structure Center. Thank you.
Thank you for that interesting and provocative talk. I'm going to take my chair privilege and ask you the first question.
Okay.
So in your opinion, how often do we need to get revaccinated?
That really depends on how our immunity decline. I think we are not certain at what level of immunity will protect us. So for the sake of current knowledge about this COVID immunity decline, I think once a year would be minimal, or maybe the maximum, hopefully.
Other questions?
Spectacular, beautiful work. That collection of sera that you have is amazing. I'm just wondering, I assume these antigenic maps are done with people that are relatively healthy, that can mount a response. Clinically, what we see nowadays with vaccination and everything is the more devastated presentation in less healthy people. So have you had the chance to study, for example, serum or antigenic map in people who are immunocompromised by any reasons, like steroids or transplant or cancer, and how those maps vary from the healthy people?
Great question. The answer is we have not. So our focus has always been pick the best among the best to study their excellent immunity, but this is a great question. So we should be thinking and dive into that immune compromise and weakened immunity patient. Thank you.
Fantastic presentation. Really, really enjoyed it. I have a question which may be a little bit out of the scope of your presentation, but if I understood correctly, letting the immune system do its job, so in healed patients and in addition to vaccination, even with not up-to-date vaccine, seems like the best course. But it may take a little bit longer. And so do you have any concerns about the impact of long COVID if you let that happen? Is it something that we should be concerned about?
Yes, great question. I think I'm only touched on the antibody aspect of COVID. So whether that itself going to help us to protect the sickler with a long COVID is another question. So I have to mention this is a narrow area of focus, but what's the implication to the broad spectrum of long-term COVID need to be better studied. But I have to mention though, since Tracy is here, we have this influenza vaccine every year, right? So if you apply the same technology to study this longitudinal influenza immunization, that would be amazing to study where the cross-reactivity is, where the protective antibody is, whether we are able to generate a new type of immunity towards newer strains of influenza.
All right, let's thank this morning's speakers, and we have a coffee break.